Okay, welcome back everyone. So in last video lecture we have seen DNA packaging, right? Okay. Then we need to discuss the scientists, various experiments the scientists perform to prove that DNA is a genetic material, and we have already seen the characteristics of genetic material, right? In our first video. So first experiment was done by Griffith. We have discussed. He took mice and then he took Streptococcus pneumonia, the bacteria that causes pneumonia disease in living organism, right? It was performed in the year 1928. So we have discussed, right? So in his last experiment, he found out that when the heat kill S3 strands was fused with R2 strands when injected to mice died. It was supposed to live, right? But mice died. The reason behind that the S3 strand must have transferred something into the R2 and it resulted into the formation of pathogenic. Okay, so continuing our experiment, so that was the experiment performed by Griffith. Now the next experiment was performed by three scientists. So continuing the Griffith experiment only, they use some additional thing. Okay, so today we're going to discuss second experiment which was performed by three scientists: Avery, MacLeod and McCarthy. Okay, so the three scientists again perform another experiment with respect to Griffith experiment. So this experiment was first performed in year 1944. Okay, 1944. So similar experiment these three scientists performed, only the difference is that they added some additional thing, like they added some enzyme. Okay, so they just continue Griffith experiment. So towards the last part of the experiment, what Griffith did? Griffith fused heat kill S3 strains. Now what is strains again? See, don't get confused, okay? The name strain is that if we keep any microorganisms on a petri dish plate or in a test tube, we name it as a strains. Okay, that background news is really disturbing every day, right? Okay, if you keep any microorganism, if you do some research in lab, suppose we want to keep any microorganism in a test tube and we have particular plate like structure, we call it as a petri dish, we call it as a strains. Okay, now if you have many plates, like for example we have 10 or 20 or 30 plates, we give them a name: S1, S2, S3, S4. Okay, so based on that, which plate shows better mode of experiment, or if we find any microorganism in any plate that shows better mode of experiment, we select that particular plate. Okay, so strain is nothing but something when we keep anything in a test tube or plate, we call it as a strains. Okay, so he heat kill S3 strain and fuse it with R2 strain. So this is a Griffith experiment only. They just continue Griffith experiment. Okay, heat kill means they heated in a fire for some time. Okay, when this was only used, heat kill S3 strain, they found out that the mice live Okay, the mice live. That means the mice live. And when Griffith used R2 strain, the mice live again, because R2 is virulent, okay, pathogenic, they causes disease. S3 is, oh sorry, R2 is non-virulent, they do not causes disease. Sorry, R2 is non-virulent, they do not cause any diseases. So he found out that the mice live. S3 is a virulent. When only S3 was injected into mice, they found out that the mice died because they are virulent, they are pathogenic, they cause disease, right? That's why mice died. But when they heated the S3 strain, they heated, okay, they found out that the mice live. That means when they give some heat, the thing which was there, that is disease causing molecule, it got burned maybe. So that's why it become non-functional. So mice found out that it lived, right? So when these two combine and injected into mice, I'll directly write like that. When these two things are combined together and injected into mice, found out that the mice survive or mice live. Mice found out that mice live. It is supposed to be, oh sorry, mice died, not live. Okay, when these two things are combined and injected into a living mice, they found out that the mice died. It was supposed to live, right? Because this was also heat kill and this is also non-virulent. We should get the mice, survive, but the mice died. They found out that the mice died. So from that experiment, he just thought that the heat kill S3 strain must have transformed or must have transferred something into the R2 strain. Okay, must have transferred something into R2 strain and transform R2, which was non-virulent, into virulent. As a result, mice died. So that was the Griffith experiment. So this experiment continued by Avery, McCarty and McLeod, and only to see that what are the things that is transferring into R2 strain. Strain means there is a cell, definitely. So what is the thing that is transferring? Okay, to see that these three scientists, they applied three enzymes. Okay, but the Griffith he could not show that what was that molecule that is actually transferring. Okay, he just came to the conclusion that something has been transferred. Okay, but it was unable to visualize what are the molecules. So these three scientists added three enzymes. They add three enzymes. The first enzyme, they used protease enzyme. Protease enzyme. The protease enzyme is the enzyme that degrades protein. Okay, this enzyme helps in the degradation of protein. Okay, this enzyme protease, it degrades protein molecule. So remember. It degrades protein molecule in nature. So when these three scientists injected protease enzyme in the mice only, okay, they found out that there is nothing changed. Mice still die. Okay, when protease enzyme was injected, after injecting these two in a mice, when protease enzyme was injected into the mice, try to understand. Okay, try to understand. When after injecting these two into a mice and then they injected protease enzyme into that mice, they found out that the mice still die. Okay, mice still die. They did not live. Mice still die. That means it is not a protein. From that they come to the conclusion that it is not a protein. Okay, so after injecting protease, no changes were occurred. Means the mice still died. Okay, they mean it is not a protein. Second enzyme, so that is not a protein. So he found out that that is not a protein that has been transferred. Okay, then they have injected RNase enzyme. RNase is an enzyme that degrades RNA. Degrades means they break. Okay, they break down. So they degrades RNA. So they injected RNase enzyme. Still, they found out that the mice still die. The mice died. There is no any changes occurred. They found. So from that, they come to the conclusion that it is not a RNA which has been transferred. Okay. Then third experiment, they use DNase enzyme. DNase is an enzyme that degrades DNA. Okay, it breaks down DNA molecule. Whenever we want to break a DNA molecule, we use this enzyme, DNase. So then they have found that when DNase enzyme were injected, they have found that the mice live. So they have found that some changes occur. Changes means the mice, they found a leaf. So from that experiment, they have come to the conclusion that that is actually a DNA which has been transferred from S3 strain into the R2 strain, resulting R2 strain into a pathogenic. Okay, and this pathogenic DNA has been transferred into a living mice. Now, when they have understood, when they have injected DNase enzyme, that they found out that DNase is an enzyme which degrades DNA. So when they injected DNase enzyme degraded DNA, they break down the DNA molecule. So ultimately, the pathogenic DNA wouldn't able to cause any diseases in a living mice. So they found out that the mice live. Okay, the changes occur. So the mice live. They have found out that the mice live when DNase enzyme were injected. So from that point of view, they have come to the conclusion that DNA is a genetic material. Genetic material means that is transferred from one body to another body and so on, generation to generation. Okay, that is transferred from one body to another body. Okay, so in exam sometimes question come from this experiment. See, the actual genetic molecular structure of DNA was given by, not structure, the actual genetic material of DNA, was given by Avery, McLeod and McCarthy. These three scientists gave the actual idea that the DNA is a genetic material. But Griffith just gave us some clue that something is transferred. Okay, he only gave us some clue. Okay, but it has a very importance because it was the first experiment in order to prove that the DNA is a genetic material. Okay, so he just gave us some clue. So from that clue, these three scientists work out again, continuing the Griffith experiment. They just added 3 enzymes to see what is that molecule has been transferred. Okay, so first they use protease enzyme. Protease is an enzyme that degrades protein. Okay, so they have found out that the mice still die. There is no any changes occur. That means the protein is not the molecule that causing disease. Okay, then they have injected RNase enzyme which degrades RNA, and they have found out that again the mice still die. That means the RNA is not the genetic material, it's not the molecule that has been transferred or it is causing disease. Then they have injected DNAs. Now when DNAs were injected, they have found out that the mice live. So that means DNase enzyme must have degraded DNA molecule. So DNA molecule has been degraded. So the mice did not receive any pathogenic DNA from R2 strain because it has been degraded already by DNase. So that means from that experiment they have concluded that it is a genetic material. Who? DNA. Okay. So DNA is a genetic material. So it was discovered by or given by Avery, McCarthy and McLeod. Remember these three scientists' name. I hope you understood. Okay. So again we have one more experiment to prove that DNA is a genetic material. Genetic material means any molecule which transfer from one body to another body. So there is one more experiment, okay? So three scientists experiment we need to study. One is Griffith, it is the initial experiment, and another one is the Avery, McCarthy and McLeod. Then another one, the third one is Hershey and Chase experiment. But Avery, McCarthy and McLeod was the first scientist who gave us the idea that the DNA is a molecule that is transferred from generation to generation or it is a genetic material. So number three we will study Hershey and Chase experiment. So Hershey and Chase experiment we need to see. Okay, this is also important. So question may ask from any three of these for 3 marks. So explain it in a nice manner, beautifully, write down your sentence, okay, along with the diagram. Diagram is not there, the experiment one you need to show. So here we have a small diagram we need to draw. So Hershey and Chase experiment, it was first performed in the year 1952. Okay. So he also proved that DNA is he means Hershey and Chase. These two scientists also proved that the DNA is a genetic material. Okay. This experiment is to confirm. Okay. Avery, McCarthy and McLeod, they have said that it is a genetic material, DNA. So to confirm that, these two scientists again prove another experiment. What they used in his experiment, they used there's a lot of noise going on in my surrounding, so it really disturbs me, but we have to carry on what to do, right? So they use one bacteria and one virus, one particular virus and one particular bacteria for his experiment. All the scientists are men, there's no woman. So they have used a virus known as T2 bacteriophage. T2 bacteriophage, the name of the virus. This is a particular type of virus, they infect bacteria. Okay, they infect bacteria only. Remember, in entrance, it is very important. So remember this thing. This is the only virus that infects bacteria. Bacteria को attack करते हैं. And they use bacteria E. coli, Escherichia coli, in short E. coli. So E. coli is the bacteria which get infected by this virus, T2 bacteriophage virus. ठीक है? You might have seen the structure of T2 bacteriophage. So I will draw here. So for his experiment, they have used one virus that infect bacteria. We call it as a T2 bacteriophage, and they use the bacteria which get infected by the virus E. coli, Escherichia coli. This bacteria is very important in biological studies. Okay, genetical studies. It is bacteria. Always researches is done based on this bacteria only, E. coli. So let me show you a quick diagram of the virus T2 bacteriophage. See, virus body is made up of only two things: protein and a nucleic acid. Nucleic acid can be either DNA or either RNA. In some virus, they contain RNA, nucleic acid, or in some virus they contain DNA. Okay. So they have a head region, and they have like this. You might have seen this type of structure in some movies or in some books. Okay, just to moves like that. This is tail. This part we call it a tail region. They have a nucleic acid inside, so it can be either DNA or RNA. Okay, so T2 bacteriophage contain DNA. Okay, DNA. So bacteria body. This is head region, we call it a head. So I'm just showing you a rough diagram, okay? So bacteria body is made up of two things only. Nucleic acid, so this nucleic acid is DNA in case of bacteriophage, and the cover layers of nucleic acid is called its protein. It is made up of protein, we call it as a capsid protein. This word, you are not allowed to forget this word. You are not allowed to forget this word. You can forget your hometown, you can forget your everything, but you are not allowed to forget this capsid. What is capsid? It is a protein cover layer of bacteria. Bacteria body is made up of protein and nucleic acid only. Okay, so the protein layer is known as capsid. तो प्रोटीन को हम लोग कैप्सिड भी बोलते हैं, कैप्सिड बोलते हैं in case of virus. So this is the diagram of virus, okay, bacteriophage virus. Now this virus, they infect bacteria. Which bacteria? E. coli bacteria. Okay, suppose here we have E. coli. I'm just showing you a rough diagram. So this is E. coli bacteria. Okay. Now when this bacteria, when this virus infect E. coli, what they do? Through these pores. Can you see these pores? Zoom it, these pores. Okay, through these pores, they inject their nucleic acid, that is DNA. They inject their DNA. Okay, so they will inject their DNA inside the bacterial body. Okay, so green in color here, it is bacterial virus DNA, T2 bacteriophage DNA. Okay. So then this DNA will reproduce inside the bacterial body. Okay, so that's how this virus infect bacteria. Okay, this is the basic things. This is the process that happens when virus infect bacteria, that is E. coli. Okay, so first is they come and bind at the surface of the bacterial body. We call it as a adsorption. It's a process of infection of bacteria. Okay, first it is adsorption, AD. What is the difference between absorption and adsorption? Anything that is adsorbed on the surface is called as adsorption. Surface में अगर adsorb करते हैं, then we call it as adsorption. ठीक है? So the first we come and bind at the surface of the bacterial body, so adsorption. Then they will inject. Spelling inject. So they will injection. So the second step will be injection. Okay, injection of what? Injection of their DNA. So they will inject their DNA inside the bacterial body. Okay. Then next, what happen? Replication will happen. Replication means this DNA will replicate their DNA inside the bacterial body. Replication means what? Making copy of their own. This DNA will make copy of their own. Okay, just like Xerox copy. So they will make copy of their own inside the bacterial body. So this is the process that takes place when virus infects E. coli bacteria. Okay, understood? Okay, then we can also write one more point: lysis. Lysis means, see, bacterial body has already its own DNA inside. Bacteria ka apna DNA already hai, right? Let me draw with black color here. In black color here, it's the bacterial DNA, okay, and the green color DNA is the viral DNA. So what happen next? The viral DNA will try to break the bacterial DNA, okay, and they will multiply. They will multiply inside the bacterial body. Okay, so lysis we call. Lysis means breakdown. Breakdown of what? Breaking of bacterial DNA. So viral DNA will try to break the bacterial DNA, but this bacterial DNA will also try to break viral DNA. This process we will see in biotechnology chapter. Okay, when any foreign DNA come inside the bacterial DNA will try to break its DNA, foreign DNA, because they don't want any other foreign DNA. Okay, so they try to break. The bacteria will try to break actually. The bacteria will try to break the foreign DNA. While doing so, they produces one enzyme, restriction endonuclease enzyme. Okay, we will see that in biotechnology chapter. But here, stick to that. So, till now what happened? The first of all, there will be adsorptions of virus, okay, at the surface of the bacteria, and then they will inject. So, injection will take place. They will inject their DNA inside the bacterial body. Then the replication will take place. Replication means the viral DNA will replicate its own DNA. They will multiply its own DNA. Then the lysis. Lysis means breaking. Okay, breaking. So the viral DNA will try to break the bacterial DNA so that they multiply their own DNA only. Okay, so this is the process that takes place. These things are happening. Okay, happening means it happens when this virus infects E. coli. So now coming back to Hershey and Chase experiment. What they did, Hershey and Chase experiment, they used two radioactive element. Okay, so they used two radioactive element. You know radioactive element in chemistry you have already studied. I know. Radioactive element is the element, you know, which helps us in visualizing any cells or any other molecule. Okay, it helps in visualization. Radioactive element. So they use two radioactive element. One radioactive element they use sulfur 35. Okay, sulfur 35 radioactive element. So sulfur 35 radioactive element, remember, it always binds with protein molecule. Okay, it always binds with protein molecule. And another radioactive element they use phosphorus 32. So sulfur 35 and phosphorus 32 radioactive element they have used. Remember, phosphorus 32 element always binds with DNA. It always binds with DNA. They have a specific rule. Okay. So next what they did, they took two test tubes. Okay. So these are the radio element they have used. and they have specific function. So they took, they have performed two separate experiments. They have performed two separate experiments. So in first experiment, so this we have a virus. So they have they have performed two separate experiments. Okay, so like that. Here we have E. coli. Here we have DNA. Here we have E. coli. So okay, let me make. So this is first experiment. Here we have another experiment. Here we have E. coli. This is your first experiment, this is your second experiment. In first experiment, they have injected this radioactive element, and this radioactive element, what? Sulfur 35. And sulfur 35 always binds with protein. So that means this is protein only, the body part, the head part is protein. So they injected here protein, that means radioactive element, sulfur 35. And sulfur 35 always binds with the protein, okay? And they allow them to allow the virus to infect the bacteria. They allow the virus to infect the bacteria along with the injection of sulfur 35, and sulfur 35 always binds with protein, not with DNA. So they have done this experiment in a test tube. Okay, here we have test tube. So next what they did, so here inside we have virus and E. coli. Okay, you try to draw a beautiful diagram. So here we have sulfur 35 along with it. Now what is the role of radioactive element? It helps us in visualization. Okay, visualization. Because these particles are very small, minute microorganism. Okay, we cannot see with our naked eyes. We need a microscope. So to see where these cells are there, we use radioactive element. You know, it helps us visualization. It gives some color, pinkish color. It gives orange color. Okay, this radioactive element. So next what they did? They allowed them inside they allowed the virus to infect E. coli inside the test tube, and then they centrifuge it. They centrifuge it. Centrifuge is kind of instrument. When you keep test tube, they used to shake like that. They shake. Okay, they keep shaking. Okay. So it is kind of instrument, centrifugation we call it as an instrument. When you go to higher study level, when you do any research work, so there will be an instrument called centrifugation. In centrifugation, test tubes are kept, and when you switch on the switch, then it used to shake. It's a shake hote hai, okay? So when this after shaking of test tube, again I need a space. The boards become very small to me. After centrifugation, see the size of the bacteria is bigger as compared to the size of the virus. Okay, because the size of the bacteria is bigger, they come down at the bottom of the test tube. So let me just draw one only. So the size of the bacteria is bigger, so they come down at the bottom of the test tube. Here, so this region, the bottom of the test tube. The bottom region we call it as a pellet region, okay? Pellet. इसको मैं रफ कर देता हूँ. I need a space. So the bacteria come down at the bottom of the test tube because it has a bigger size as compared to the virus, and the bottom region of the test tube we call it as a pellet region. And the virus, it remains here. Okay, because it has a bigger size, and this region This region we call it as a supernatant region. Supernatant region. Now, next they have found out that this radioactive element, that is sulfur 35, in the viral body only, virus body only. They did not find in the bacteria body. So when they visualize the test tube, they found out that the sulfur 35 still stuck with the viral body with the protein. They did not transfer into the bacterial body. Okay, when they visualize, they found out that the sulfur 35 is still with the viral body. They did not enter into the bacterial body. So from that experiment, the first experiment, they have found out that the protein did not transfer from one body to another body. Okay, so this was the first experiment. The next experiment, similarly, similar experiment was done. The only difference is that instead of sulfur 35, now they injected phosphorus 32 radioactive element. Okay, so instead of sulfur 35 which binds with protein, they injected phosphorus 32 element. Now phosphorus 32 element always binds with the DNA molecule. Okay, it binds with the DNA molecule. Now similar experiment was done. They centrifuged it. After centrifugation, okay, I'll directly show this part. Okay, so after centrifugation, here the size of the bacteria are bigger as compared to the size of the virus. They come down at the bottom, the virus remain at the middle because the size is bigger. The region where the virus are located, we call it as a supernatant region. Supernatant. And the region, the bottom of the test tube, we call it as a pellet region. Okay, there is a name given. We use the name pellet region and supernatant region. So here they have found out that the DNA which was injected into the virus, that DNA was found inside the bacterial body. That DNA was found inside the bacterial body along with the phosphorus 32 radioactive element. Phosphorus 32 radioactive element. Now when they visualize this test tube, they have found out that the radioactive element phosphorus 32 which was injected into the viral DNA, this radioactive element was found out that inside the bacterial body. So from this experiment, they have come to the conclusion that it is a DNA that is transferred from one body to another body, and this proves that DNA is a genetic material. So this experiment 100% confirms that the DNA is a genetic material. So for this experiment, what they use a virus that infects bacteria. We call it as a T2 bacteriophage and the bacteria E. coli. So they have used two radioactive elements, sulfur 35 and phosphorus 32. Sulfur 35 always binds with the protein part of the virus. So in first experiment, they have injected sulfur 35 with the protein of the virus, and they have found out that after centrifugation, they have found out that the sulfur 35 is still there with the viral protein. They did not transfer into the bacterial body. Okay. The next experiment, they have found out, or they have injected phosphorus 32. Phosphorus 32, it binds with the DNA of the virus. They centrifuge it. After centrifugation, they have found out that the phosphorus 32 radioactive element inside the bacterial body. So from this experiment, it is proof that the DNA is a genetic material and it is transferred from one body to another body. Okay, so that's it. That is all about Hershey and Chase experiment. We have completed 3 experiments. Study all. Out of these 3, any one of them may come. Okay. So next class we will see about the RNA.